Okay, now you can start.
Okay, sir. Thank you, sir. Good morning, everybody. I hope I am audible.
Yes, yes.
Yeah, good morning, sir.
Thank you, ma'am. Thank you, sir. So I am very happy today that in our PhD webinar series, we have our 11th consecutive speaker, Dr. Madhumala, with us. And before introducing her, I would like to invite Dr. Arvind Deshmukh to speak few words.
Thank you.
Thank you.
Thank you. Thank you, Jay. I'm very happy today. This is a 11th lecture of PhD student. And all these are fresh PhDs. So all attendee will get the recent knowledge in the subject. And we want to encourage, we want to inspire young generation for doing PhD and for doing research in India. Ultimately, we want to serve India. And today I am very happy that many PhDs and many senior persons, including Dr. Aditi, Dr. Tunisha, then Dr. Swarnajit Singh, Dr. Rohini, Dr. Ashwini, and many others from India and abroad. Some are from abroad, like Namdev has joined from abroad. So people are attending and encouraging, inspiring the speaker. I am thankful to all attendee and Gujarat unit for attending, for organizing this seminar. Thank you. Thank you very much.
Thank you so much, sir. Thank you. Madhumala ma'am, are you ready? Ma'am, please check if you can share your screen.
She can share.
Okay.
Madhumala ma'am, try to share the screen. Your course now.
Ma'am, if you are facing any issues, we can share the PPT for you. Just let us know.
She is muted.
I guess so. Dr. Meenal Trivedi, Gujarat State MDSI President, has sent her regards for our lecture. Now, in the interest of time, I think I would like to introduce the speaker, Dr. Madhumala Y. Ma'am is currently working as Assistant Professor, Department of Biotechnology, Basaveshwar Engineering College, Bagalkot, India. Ma'am has done her MPhil as well as PhD. I would like to mention that Madhumala ma'am has guided 21 UG students and 6 PG students in their dissertation work. Ma'am's research interest area are bioremediation, microbiology, and pharmaceutical. Madhumala ma'am has received funding for 6 different projects from the various government and private agencies. Excuse me, I would ask everybody to please keep yourself muted. Madhumala ma'am has published 6 national and international peer-reviewed research papers in various journals. One very important thing I would like to mention here, Madhumala ma'am has attended 48 different conferences, symposiums, workshops, and training programs. Before that, Madhumala ma'am worked as a senior research fellow in DBT ad hoc funded project entitled Biodiversity of Bacteria in Western Ghats for a period of 2 years. Other than that, ma'am has also worked as a guest faculty in BEC, BGK from 2006 to 2007. Now I would like to welcome our today's speaker, Dr. Madhumala. Madhumala ma'am, are you able to share your screen?
Yes, sir. Can you tell me the mail ID? I'm unable to share.
Okay, I think you are
Hello.
good. So I think it should be doable. Okay. Can you tell us what is the error or issue you are facing?
Like I'm unable to connect my mobile laptop. I don't know what is wrong actually.
Okay.
So if you write,
Okay.
send your mail ID.
I think there is some connection issue. Your voice is
What I will suggest, Madhumala ma'am, you close
Yes,
your
sir.
laptop, open your mobile only, join with the mobile, and Jay will share
sir.
the screen.
Sure, sure. We will do that.
So you join with the mobile, which is more easy.
Yes, sir.
Close
Yes,
your
sir.
laptop.
Like, yes, sir. I have joined with my mobile.
Jay, you can share the screen.
Sure, sir. I will do that. I will just share my screen.
Tell me the mail ID, sir.
I will just WhatsApp.
Yes.
Deshmukh, sir.
Yes.
There is one more account.
Jai,
Can you make that co-host, please, so I can share my screen?
Jay, you can share the screen. Screen is not shared by anyone. You can share the screen.
Sir, I request you that there is one more account, Jai Jatia, in the participants. If you can make that account co-host.
Okay. I will. Just 1 minute.
Thank you.
Yeah. Ahmed. Okay.
Okay, sir.
Ahmed. And I request all others to mute. Please mute your mic. Everyone.
19.
And Madhumala ma'am, close your laptop.
AHIRJAY19@gmail.com.
As soon as I receive the PPT, I will share.
Yes. They are sending by
Sir, I
mail.
have sent.
Okay. Okay, I'm waiting.
So, ma'am, you start your talking. In the meantime, he will share the screen. Dr. Madhumala
Yes,
ma'am, you start. Please go ahead.
Good morning. Yes. Good morning.
Yes, we can listen you.
Yes, sir. Okay, my research topic is biofilm induced changes in reduction of hexavalent chromium by indigenous microbes. So this is my title. So I'm mainly worked on environmental microbiology, and my guide is Dr. Veena Surgavi and Bharati Meti. I've done it in VTU. So this, the main aim of this is I just wanted to mainly go with the bioremediation techniques, which are more helpful in cleaning up the environment. So that's I was very much interested in bioremediation. So I have selected this topic. An introduction already Jay sir has given about the introduction. To start with, sir, shall I start once the slides are open?
No worries, just a second.
Yes, sir. Okay, indeed, but I would like to tell why I chose top. One thing is, mainly the hexavalent chromium is going to be one of the elements which is going to be very toxic and carcinogenic. And the thing is, I have utilized the indigenous microorganisms here. So indigenous microorganisms are the ones which are found at the site where the contaminants are there. So here I have selected the sites which are contaminated with chromium and have isolated the organisms from these sites. And I've also used the biofilm forming organisms, which normally are the ones which go for biosorption. So the biosorption is the process which is used here for reduction of hexavalent chromium. Jason, did you get the PPT?
Sorry, ma'am, I am unable to hear. Do you want me to change the slide?
Did you get the PPT? I'm unable to see the slides actually.
Oh, I think I have shared the slide. Can anybody confirm that my slides are visible?
Yeah, slides are visible.
Your slides are visible.
Ma'am, I suggest that you open your slides in your PC and I will simultaneously share.
Yes, sir. Yes, sir. Yes, that's better, I feel. Let me go with the introduction. Okay, first I would like to know the chromium properties. So chromium is one of the elements which has a symbol Cr and atomic number 24. It is the first element in the group 6b, which is steely gray, lustrous, hard and brittle metal. So this actually has different oxidation states ranging from 3 to 6. So the chromium compounds are very stable, especially in the trivalent and the hexavalent state. So this trivalent is one of the essential nutrients for maintaining the blood glucose level and also for the metabolic activities. But hexavalent chromium, it is one of the very toxic form. It is actually used in the anti-corrosive. It is generated during the welding of the stainless steel or the metal structures coated with a chromate paint. Next slide, sir. And the products which contain the hexavalent chromium are the pigments, paints, inks, and the plastics, anti-corrosion coat. It is used as an anti-corrosion coatings. It's also used in the alloys, in the textile dyes, and it's also used in wood preservation and tanning of the leather. There are different forms of the hexavalent chromium, like lead chromate, zinc chromate, barium chromate, calcium, potassium dichromate, all these forms of the chromate are the forms of hexavalent chromium. Next slide, sir. These are different forms of hexavalent chromium. So let us see the health effects of this. A lot of ingestion hazards. It is erosive to the stomach, and it also causes the hemorrhage and sometimes death. It normally affects the eyes. Those who comes in contact with the direct eye contact, especially with the chromic acid or the chromate dust, that can cause a permanent eye damage. It also affects the respiratory tract because it's a very fine element, and during the process, a lot of this fine element is being it enters into the air, and it irritates the nose, throat, and the lungs. And repeated or the prolonged exposure also leads to the damage of the in the ulcers. Prolonged leads to the ulcers.
Excuse me, ma'am. I think some issue with audio, I guess.
Yes, ma'am.
Yeah, just a minute. Okay. So the prolonged skin contact can result in dermatitis and skin ulcers.
I think the voice is better than before.
Okay, sir. So this, some of the workers, they develop an allergic sensitization to the chromium, and it also can cause a serious skin rash and ulceration, and even it also leads to occupational asthma and perforated nasal septa and pulmonary congestion, liver damage, and even the perforated eardrums may be the result. And it also may lead to edema, epigastric pain, and discolorations of the teeth. And based on this, next slide, sir.
Yes.
So the National Toxicology Program in 2010 that hexavalent chromium in drinking water showed the clear evidence of carcinogenic activity, especially when it was tested in the laboratory animals. It also increased the risk of gastrointestinal tumors. And even exposure also has been linked to immunity disorders, neuropsychic neurodegenerative disorders. Mainly, the zinc chromate is one of the most potent carcinogen. So these are all the different types of ill health effects being found due to the exposure to the chromium. And there are different methods of chromium reduction. Normally, there are the conventional methods which have been found. The conventional methods are ion exchange, filtration, precipitation, electrochemical precipitation, chemical reduction, adsorption, membrane technology, evaporation recovery, and these are some of the physical and chemical methods normally used in the chromium reduction. And another method, the most suitable method, I feel, is biosorption. Biosorption is a mechanism which mainly consists of ion exchange and adsorption and diffusion. So the adsorption and diffusion, especially through the cell walls and cell membranes. Biosorption is nothing but the adsorption using the biological Biosorption is the process of adsorbing the different metals and components using the different biological components in a liquid solution. It can be removed easily, and it's mainly used for immobilization and sequestration of various metals from the effluents. And by why do we use these microorganisms? We can use the different biological components, of course, for biosorption, but using the microorganisms have a great advantage because they have a high surface area to volume ratio. And they can also provide a large contact interface which would interact with the metals from the surrounding environment. Next slide, sir. And coming on to the biofilms. So the biofilms are defined as the communities of microorganisms which are attached to the surface, and the interactions with the biofilm can be a bioaccumulation or oxidation or reduction, or even the involvement of the enzymes by, in order to go the extracellular precipitation by the metabolites which are produced by the bacteria. And it also includes the biological transformation of hexavalent chromium to trivalent, which is the novel one or the recent one. Here, the hexavalent chromium serves as an electron acceptor for the oxidation of organic compounds. And there are different types of bacteria which can reduce this chromium to trivalent state. As I told you, the hexavalent is a carcinogen, but the trivalent is found to be the very less toxic, and it is the one which forms the precipitate. And easily can separate the liquids from the hexavalent chromium is greatly soluble in the water, and it is not easily removable. So once you convert it, transform it into the trivalent, it is easy to remove. So there are different bacteria which have the capacity to go for biosorption or transformation of this to trivalent by using 3 different techniques. One is they can go with the redox reactions, or they can go with the adsorption metabolic reactions on the surface. And the third one is intracellular precipitation. In the some organisms which have so these have been reported to have the enzymes which are responsible for reduction of hexavalent. So next slide. So the biofilm formation, you can see the image here. So this image will show you that the planktonic cells are found to
Excuse me, ma'am. Do you have an opportunity to connect with another network and speak? Hello, Madhumala ma'am.
Yes, sir. Yes, sir.
I think there is some issue in the voice. So do you have an opportunity to connect with another network and speak?
She is having network problem. Network is not strong.
Ma'am, we are not able to hear you. I'm so sorry. Sir, do you have any suggestions?
Ma'am, you join with other mobile. Your colleague's mobile, any other mobile. You transfer this link in another mobile and join with other mobile. Madhumala ma'am, the voice is breaking. The mobile quality is not good. Internet quality is not good. So you join with other instrument. Jay sir,
Yes, sir. I think
you
I will
call her.
Yes, I am going to do that.
You call her and tell any other mobile.
I will. Just a moment.
So in the meantime, I will make an appeal to all attendees today. I welcome all attendees. And this is our lecture series. Fresh PhD students, not completed more than 3 years. What we are doing means if few of you are there, please you can join our lecture series. We are giving memento. Now Madhumala has joined with another instrument, I hope so. Madhumala ma'am, can you listen me?
Sir. Yes, sir, I can now, sir.
So you start your lecture.
Yes, sir.
Yes, proceed.
Yes, sir. So the biofilm formation. So the biofilm formation takes place by the normally the planktonic cells, they go on getting attached one above the other on the surface, and as the colonization takes place, then further once the colonization has taken place, it grows completely to form a mature biofilm. You can see in this picture that the mature biofilm has been formed. Once the mature biofilm has been formed, the microorganisms move outside. So this is how the biofilm formation goes on happening. So normally the biofilm is an advantage and disadvantage both. So here in the bioremediation, it is found to be more advantageous, but actually in the medical field, it is found to be more disadvantageous because it is the one which normally causes the dental plaques, and it is also one of the things which normally causes rusting of the iron and many other type of disadvantages. Okay. Next slide, sir.
1 minute, Madhumala ma'am. You can start your video also. You start your video also.
Yes,
just
sir.
Start your video.
Yes.
Okay, you can start. It's a very simple. Your video is closed.
Yes, sir. That's what I'm checking, sir. Yes, no. Yeah.
You are joined by mobile, na?
Yes, sir. I've joined. I have joined to another mobile, sir.
So
Yes,
at the
problem.
bottom, there is a video option. You just start the video.
Video.
Okay, if not possible, you can continue.
Yes, sir. Yes. I'll try to do in meantime, sir. So the main Sir, can you skip the slide?
It is visible. It is visible to us.
Which slide you want
Yes.
to, ma'am?
This you skip the slide, sir. Next one. Next one, sir. Coming to the objectives. Skip 2 more slides.
Okay, ma'am. I'm on that slide.
Yeah. Right. So the objectives of the main study here is to reduce the hexavalent chromium to trivalent using the bioremediation process by using the indigenous microbes. So I suppose everyone knows that the indigenous microbes are the organisms which are normally got from the source, from the contaminated sites, so that you get these organisms have the capacity to stay with those things and also reduce the same. So we have selected some of the sources like normally the electroplating industry, tannery waste, and the municipal waste, leachate, and the cement industry. These are all the things where the hexavalent chromium is found to be one of the waste which is normally found. So we use these sources to screen and identify the microorganisms which can reduce the chromium. At the same time, we also screen these organisms which can produce only the biofilms. Okay? And once we have gone with that, the another next objective was mainly to optimize the parameters, growth parameters, so that we can produce the proper biofilms, and also optimize the parameters which affect the reduction of hexavalent chromium. So we also have involved the design of experiments using the different variables, so that this design of experiments have helped us to go with the proper optimized conditions for the reduction analysis. Next slide, sir. And also we have gone for finding out the enzymes which are responsible for reduction of hexavalent chromium. So coming to the methodology. So this is our experimental plan. First, collecting the sample, that is from the source. We collected the different samples and then isolated the chromium reducing microbes and screened for the biofilm producing microbes among the one which reduce the chromium. They were both biofilm and non-biofilm formers, but we selected only the biofilm ones, and then gone with the biochemical characterization and study of the growth curve, optimization of the growth conditions by using the different parameters, and the batch studies, column studies, and also by employing the RSM studies, and some isothermal studies were also being conducted. Next, sir. So the isolation methods were, as I told you, these were the samples which were being collected, the tannery effluent, soil samples from the electroplating industry, leachate, cement industry, and sewage sample. And we used the serial dilution techniques, and we employed the hexavalent chromium in the sample while we went for the pour plate method. Along with the media, we also introduced the hexavalent 1000 mg of the hexavalent chromium, so that the organisms which could tolerate only could grow. Next slide. So these are the effluents which were being used. There's only two pictures I have put. This is from the first one is from the leachate, and the other one is from the tannery. Next slide. And these are the plates with the isolates where we have followed the first we followed the 4 plate, then we had gone for the streak plate and the spread plate for mass multiplication. Next, sir. So later on, once we isolated the organisms, so these were the isolates there were different isolates. So once we got the isolates which could tolerate the 1000 mg concentration of hexavalent chromium, then we tried to screen the organisms which produce the biofilms. Next slide, sir. So isolation and characterization of the biofilm forming organisms. So we use the 3 different methods. That's the Conger agar method, tube assay, and the microtiter plate assay. So next, so the Conger agar method was not so effective. This is the tube assay method. So in the tube assay method, we inoculate the organisms in the tryptico soya broth, where these organisms produced a film on the layer. So if you can see, there is a film formed on the layer. So later on, this next slide, sir. Tube assay. After this, inoculating it into the tryptic soya broth, so there was a film formed. So we removed the broth and stained it with a crystal violet, and we put it upright down after removing the broth. So whatever the biofilm was formed, it adhered to the inner surfaces of the test tube, and it stayed on the thing. And this is the microtiter plate assay, where this was run on the ELISA plates. So this on the ELISA plates, so one is 200 microliters of the sample was inoculated within the wells for about 37 degrees Celsius for 24 hours. Then these wells were washed with phosphate buffer and stained with the crystal violet and read it on the ELISA plate reader. So this ELISA plate reader showed us the different readings at 540 nanometers. So the 0.2 reading gave a weak type of biofilms, and 0.2 to 5 was moderate, and more than 0.5 was found to form the strong biofilms. So next slide, sir. So this is the result what we got, and it was found that there were some excellent biofilm producers. That's isolate 1 and isolate 7 formed a very good excellent. Isolate 1, 3 and 7 formed a very good biofilms, and there were other things also like 9, 10, 11, 12, 13 also formed good biofilms. Only very few formed the moderate ones. Among the 16 isolates, we removed the two things, and 14 isolates were further restored for the chromium reduction process. Next slide, sir. So this was the thing with the tube assay, where we stained with the crystal. We removed the broth and stained with the crystal violet, and all the biofilm get adhered to the surface of the inner walls of this test tube, and that indicates that the biofilm. These are all the biofilm producing organisms, and the non-biofilm doesn't form any layer as such. Next, sir. And after finding out this, we went with the biochemical test to identify these bacteria. So like IMVIC test, indole, MRVP, citrate, gelatin, catalase, and gram reaction test were being conducted. Next. And these were the results of the biochemical test. So this is mainly to go with the phenotypic type of identification. Next. You can just go on moving. So this is the indole test, which is done mainly to find out the ability of the microorganisms to utilize the amino acids like tryptophan and produce the indole. Next. And this is the methyl red and the August Prosker, that's which comes under the MVIC. So there are different, there are some of the organisms which showed methyl red and August. Some showed methyl red positive and some showed August Prosker positive. So normally the methyl red reduction test, the one which produces the acids shows the methyl red as positive, and the one which doesn't produce acids, that's other products other than the acids, shows the VB as the positive ones. Next one. Next is next slide. Next is citrate. So this citrate is the one which can tolerate the citrate, or which can utilize the citrate as a carbon source, can utilize, and the blue color, the green color changes to blue. Here the bromothymol blue is used as an indicator. So when the citrate is utilized, it changes from green to blue. That indicates it can utilize the citrate. And gelatin hydrolysis, and some of them which are proteolytic, the organisms which are proteolytic can reduce the gelatin. It can go for hydrolysis of the gelatin. And there were some organisms which could go with us. Next. Next slide, sir. So with the Gram reaction results, we found that some of the isolates were Gram positive rods. We also got the Gram positive and Gram negative rods, and some of them were cocci. So they were rods, cocci both, and Gram positive and Gram negative rods. And two organisms were cocci. One were very small, which belongs to the Micrococcus, and another one is Staphylococcus aureus. Next slide. And these isolates were identified by using the 16S rRNA technique and the BLAST. So we found that 6 of them were Bacillus species. There are different species. Some were very novel also, like Bacillus hyanacea was one of them, which is found to be the new one, which is not commonly found in in the bioremediation. And Enterobacter hormechi is also one novel one. Lysine is one more. Acinetobacter variabilis is also not found in the bioremediation of hexavalent chromium. So these are some of the, even Micrococcus elovirae is also one of the organisms which are found to be the novel ones. And all these organisms were submitted to the NCMI. Yes. So this actually, these all isolation with the 16S RNA was done by using the Barcoding Institute of Technology with Aurangabad. So these people helped me to go with this BLAST 16S RNA technique. Next. So the phylogenetic tree of isolates, this is the phylogenetic tree which showed the resemblance with different organisms, because we had many of the 6 of them were Bacillus and others were the different thing. And it was found that these organisms were identified and were characterized. Next. So after characterization, we had been to the chromium, next slide. Chromium reduction. So the chromium reduction test was analyzed by using the different chromium concentrations from 50 mg to 1000 mg, 50, 100, 200, 300, 4 to 4, 5, 6, 7, 8, 9, and 1000. So till 1000, until in the literature review, very few literatures I found that it was still 1000. And we analyzed, we wanted to find out whether now we can reduce 1000 mg of chromium also. So we analyzed from 50 mg to 1000 mg with all these organisms. And you can see the picture here. Before growth, it is the solution is very clear. But once after the growth, it is turbid and the filmy layer is normally found on the surface because it is a biofilm producing organism. So once the growth was there, so it is normally after 24 hours, we started, before we did the analysis for chromium reduction after 6 hours, but it was not so fruitful. So we found it was fruitful only after 24 hours. So 24, next slide, sir. So after 24 hours, we did an assay which is known as diphenyl carbazide assay. So this diphenyl carbazide assay includes the diphenyl carbazide as one of the substrate which is added to the solution here. 1 ml of this solution, the broth, which was where the organ after the growth of 24 hours, 1 ml of this sample was taken. And to this, we added 9 ml of water and 0.5 ml of this diphenyl carbazide solution, and just 2 to 3 drops of H3PO4. And after this, after a reaction, it was left for about 15 to 20 minutes. The reaction took place where all the hexavalent chromium, what was there, the amount of chromium is produced, the amount of the color also increases. Here in this second part, you can see after reduction part. So the color, there is from colorless to highly colored. The as the reduction goes on taking place, the color goes on reducing. It becomes colorless once it has completely been reduced. Next slide. So these are the results of the isolates. And we have found that, as I told you, we have analyzed from 50 mg to 1000 mg per liter. So it is the isolate 1. So it has found that within 2 to 3 days, there were 100% of the reduction till 500 mg. But after 500 mg, there was reduction was lesser. So in 1000 mg, after 5 days, it was found about 58%, and further there was no reduction. That is 58% of the 1000 mg chromium was reduced. The same thing with isolate 2. It was about 56% in 1000 mg concentration, but 500 till 500 mg, there was complete reduction. And with isolate 3, it was about 85% in 600, but below 500, it was 100% within 2 to 3 days. But after 5 days, it was about 52% of reduction in the 1000 mg. So in the same way, all these isolates showed a very good result about, so it could go with the reduction about the maximum percentage of reduction was 58%, 1000 mg. So normally the reduction takes place very slowly once the concentration of the chromium goes on increasing. The lesser amount of the chromium, the reduction will be faster, but as the concentration is increasing, it reduces. So this may be because of the toxicity of the chromium which has been found, because as I told you, it is very toxic. Even the microorganisms also face the same thing. It is found to be toxic to them also. And maybe the process at the start of reduction is slower because the organisms need the time to get adjusted. So the reduction process started after 24 hours, but in the 50, 100 and around 200 to 300 also, the reduction was once after 24 hours, the reduction started, the reduction was completely 100% within 2 to 3 days. So this indicated that it took a stage the toxic and needed time for the adjustment. Next slide. So these are all the results of the different isolates. You can move on, sir. Yes. So the biomass produced in mg at different intervals at 500 mg per liter. So it found there was a the biomass increased, and later on it started reducing. That is, the biofilm the growth was at the peak till 5 days. So maybe the once after that, the biofilm mass will be there. The biofilm was completely matured after that. Till that, after that, the cells slowly started disaggregating. Next slide. And in between that, we also check for the viability, because normally the growth curve we see that is 1 or 2 days after that, normally the process goes on declining. So here we saw the viability, and it was found that the viability was there till the process ended. So we even tested for 8 days also. Every day we tested for the viability, and the organisms were viable, and they were in the good state to still reduce the chromium. Next slide, sir. And in the reduction process, we also went with the SEM analysis. So the SEM analysis, in the SEM analysis, we found that we went with SEM analysis only with 3 organisms, which were highly efficient. And one among that was Acinetobacter. So as you can see the 2 pictures here. The first one is prior to exposure to the hexavalent chromium, which are healthy cells. You can see their shape. It is very nice. And the second one is after exposure. So here the surface of the cells are found to be slightly rough, and the ridges are found on the cells due to the accumulation of the metals. And even because of the toxicity also, the cells are slightly wrinkled. Next slide. So with the same thing here in the Staphylococcus aureus, so you can see here the cells in the first one prior to the exposure, you can see the cocci very nicely. But after the exposure, you can see that they have aggregated and they have formed the biofilm layer. And these cells here, there is the rupture of the cells and the filmy layer being formed. Next slide, sir. Next slide, sir. Okay. So this is the, just a minute. So the Bacillus species, the before slide. So it is after exposure, you can see here the cells are shrinked. They are shrinked and they form a mat over there. So this is with the Bacillus species. So next we also performed the next slide, sir. So these, whatever the consequence we have found, maybe it is because of the toxicity of the chromium, which was been, which has made these cells to shrink or form the wrinkled surface, and the as they are all the biofilm forming organisms, they also form a filmy layer. And we also have gone through the EDX analysis for 2 organisms. So in the EDX analysis, we have found that the Bacillus and the Staphylococcus aureus. In the Staphylococcus aureus, it was found that the 1000 mg per liter of the chromium, whatever we had taken, had decreased to 23.55 grams. 23.55 grams. So that indicated about 97.6% of reduction. Next slide, sir. And in the next, like EDX, that's a Bacillus, EDX analysis in the Bacillus indicated that the chromium was reduced to 19.71. That is CRK here, you can see in the last column, that is 19.71. So there was 1000 mg was reduced to 19.77 mg. That indicated it was 98.2% of reduction of the chromium within the cells. Next one, sir. So this, all these SAM analysis, EDX analysis, and whatever the diphenyl carbazide assay we had performed, so those all indicated us that they were the ones which showed us about the how the reduction has taken place within the batch studies. We also performed the column experiments where we use the sand column. So normally we use the different components for reduction analysis, like cellulose acetate and agarose. Many of these things are being used, which are costly enough. So I thought of using the sand, which is also a good adsorbent, and we are into biosorption. So why don't we use the sand, which can act as an adsorbent, which also can act as a layer of substratum for a for my biofilm forming organisms, so that it can form a substrata there and go for the biosorption process. Next one, sir. So, so we found that the adsorption rate was higher, till 90% of this adsorption was being found in the 1000. We used only the 1000 mg here. So first we inoculated the in the column, we set up a column, and in the column we fed the chrome. First we fed the nutrient broth. Along with that, the Bacillus organism was inoculated, and it was incubated for 24 hours. Then the hexavalent chromium was being added there. 1000 mg of hexavalent chromium was added, and the column studies were done every 2 hours. And as the analysis was done for 2 hours, there was a reduction being found. The sand was also found to act as a sorbent, and these organisms had formed a nice biofilm on the sand in the column. So this, after every 2 hours, the analysis was, it was being studied, and it was found that after 10 hours, around 10 hours, there was about 90% of the reduction of the chromium. And these option studies were also done, where we also tried to find out the chromium being found within the sand column. Next slide, sir. So by this we also found that the sand can also be used for adsorption of the chromium along with the biofilms. Next one. So design of experiments. So we also used these design of experiments mainly to optimize the conditions for the minimization of the time and also for increasing the reduction process. So we use the 4 variables here. That's the pH, temperature, inoculum, and the chromium. So the pH used here was from 4 to 8, and temperature 30 to 40. Inoculum size was from 2 to 10, and the chromium dosage was from 100 to 1000. So here the RSM design of about 62 experiments with 4 variables at 5 levels was produced, and we found that. Next slide. So here, this is the surface plot of percentage of reduction. So here it was found that the inoculum size 7 was found to be the most suitable one for the reduction process, and the dosage chromium dosage of 550 was found to be more effective, and this was done within 3 days. Okay, so in 3 days we found that there was complete reduction, and the inoculum size of 7% and the pH of 6. Next slide, sir. Inoculum size of 7%, 7% and chromium dosage of 550 was found to be the most at the temperature 35 degrees Celsius. pH 6, temperature 35 degrees Celsius, inoculum size 7, and chromium dosage of 550 was found to be the conditions where it can reduce the chromium, 100% of the chromium in 3 days. Next, sir. Next slide, sir. So the isothermal results, isothermal studies were also being conducted to find out which were the models which could, because these were the biosorption studies. So we wanted to connect it with the adsorption, where the Langmuir model and the Freundlich models were being taken here. So we studied the Langmuir and the Freundlich model. Next slide, sir. So it was, we studied with the 5 isolates, and we found that the Langmuir and in the both with the isolate 1 and 2, we found that the Langmuir model was found to be more better than the Freundlich one. Okay? So in the first isolate, the Langmuir and Freundlich had given the R-square values more than 0.99, so which indicated, so these models actually were studied mainly for trying to find out the adsorption, what is the rate of the adsorption being found. So this indicates there was a good amount of adsorption being found by the biofilm forming organisms. Next slide, sir. In the isolate 2, actually the Freundlich did not obey much, but the Langmuir model was found to be more accurate for this. And with the isolate 3, it was found that both of the among the Langmuir was found to be more suitable than the Freundlich. But in the 4th isolate, Freundlich was found to be more favorable than the Langmuir. In the same way with the isolate 5, next slide, sir. Isolate 5, the Freundlich was found to be more suitable than the Langmuir. So these both of these isothermal models indicate that there was a good rate of adsorption being found on the surface of the media, or these are found to be more better for the reduction process. Next one, sir. Enzymatic reduction. Next slide. So the enzymatic reduction, so we also studied whether there are some intracellular enzymes which are mainly responsible for the enzyme reduction. And based on the literature survey, we had found that there are some chromium reducing enzymes which act as a, like where the copper acts as a prosthetic group and which helps in reducing these enzymes. Okay. So we try to find it out. Next slide, sir. So based on this, what we did is normally in these microorganisms, whatever the biofilm organisms are there, they produce the extra EPS, that is extrapolymeric substances. So these extrapolymeric substances, extracellular polymer substance is one of the substance which is protein rich. So we found out the, we tried to find out the protein estimation in the cell. So that is the amount of the protein before reduction or before biofilm production and after biofilm production. So it was found that after the reduction process, so the protein estimation was found to be very high. You can see here. So before, the protein estimation with the different organisms is not more than 0.5. Okay. So, but after the reduction, it is found till 0.95, the amount of the protein is found. So that indicates, that is the extracellular polymer substance is one of the substance which is being produced, which indicates the protein production there. Okay. So this is one of the things which helps us to find out that these are the substances which helps the biofilm to go for the biosorption. Next slide. So this also helped us to find out the characterization of the chromium reducing activity. So where we found that the because of this extracellular polymer substances, so there was a reduction being found. So in 2 hours, these organisms were found to, which were more, which are having the more of the protein, are found to produce more amount of the chromium. So that indicated that they can reduce the chromium faster than the one which doesn't contain the extracellular polymer substance. Next slide, sir. So coming to the conclusion, so there were actually 25 isolates being obtained from the from the indigenous sources. Among that, we found 16 strains which showed the tolerance towards the 1000 mg of hexavalent chromium, and among them 14 strains were able to produce the good biofilms and excellent biofilms in the, and we performed the reduction analysis with these 14 isolates. Among them, 5 isolates were found to be the excellent ones, which also were conducted with isothermal studies, and these were able to reduce the hexavalent chromium from 50 mg per liter to 1000 mg per liter. These organisms, with the optimization, we found that they could also grow very well at the normal room temperature. They didn't need any other temperature adjustments, like whether to go with a lower temperature or higher temperature. So normal room temperature was enough, and also the conditions like neutral pH was also more favorable for them. And aerobic condition, we need not provide any aerobic conditions for these organisms. They normally grow at the normal conditions. And these, whatever the organisms are there, they are very efficient in reducing the chromium from 50 mg to 500 mg. That is 100% of the reduction was found in 500 mg per liter. But as we increased the concentration, it was found the reduction was, the range was about 49 to 58%. Next slide. So all these isolates were proved to be very efficient in reducing the hexavalent chromium. Maybe in the higher initial concentration, there was a reduction in the percentage because of higher toxicity. Because as the concentration goes on increasing, the toxicity also increases, and the organisms may not be able to sustain it. Okay, and so the growth rate was slower, and the reduction rate also was slow, and it took more than 5 days to reduce to 58%. And these were, and as the SEM and EDX analysis also showed that they could form the biofilms, and also the formation of the ridges, wrinkles, and other things. They showed that it was all these wrinkles and ridges and variation in their size and all. It was all because of the toxicity of the hexavalent chromium. And this and whatever the optimization studies were done, that is RSM design was done. So this helped us to find out that the normal pH of 6 and about 8% of the inoculum and 30, that's a room temperature of 35 to 37 was more suitable, and 82% of it was reduced in 2 days. So this and the enzyme studies also help us to know that the enzymes responsible for reduction are from cell-free extracts. Next slide. And in the column, we found that the 0.6 mm, we used two columns, 0.3 and 0.6 mm, and the flow rate was from 10 ml to 25 ml. So these were the two variables we used here. And as we found that the adsorption rate was more in the flow rate of 10 ml per hour. So, but as we increase the rate of this, maybe the exposure to it was lesser. So the 0.3 mm with a flow rate of 10 ml per hour column was found to be more efficient in absorbing the forming the biofilms and absorbing the chromium than the 25 ml per hour of the column. Next. So adsorption isotherms, which were conducted for the Langmuir and the Freundlich. So they also indicated that the Langmuir adsorption isotherm was found to be good in some, and Freundlich was found to be good. So Langmuir adsorption, normally isotherm occurs through the monolayer exposure, and the metal binding at the biofilm surface is normally measured with adsorption here. And Freundlich is, which is suitable for the Freundlich. Here there are multiple sites, multiple binding sites. But with this Langmuir, it's a monolayer. But with the Freundlich, it is the multi-layer. So where it can bind to more layers, and it can it has more of the adsorbent surface. And some of these organisms were very efficient in showing that Freundlich adsorption isotherms were more suitable. And the microorganisms, as they were selected from the indigenous sources, maybe they were more evident in going with the bioremediation. And this process is found to be, whatever the biosorption process I have followed, is found to be more friendly, and it is what do you call it as? It is more eco-friendly process and cost effective. You're not going to spend any, because conventional methods where you go for ion exchange, chelation, and different types of separation techniques, which are very cost effective, which are very costly, and they also leave lot of wastes with a variety of chemicals being found. So with this biosorption, it is one of the most friendly process and eco-friendly process, and a less expensive. So this should be preferred more than the conventional methods. Next slide, sir. So this is my work which I have done. So some of the future work which can be done here is like kinetic studies can be done with the first and second order model, and mathematical model can be created for treating the contaminated soils. Along with that, you can also go for designing large scale bioremediation system. We had done only for the small scale here. I've not used any industrial effluent here. I've done it with a synthetic type of production I've done. Like I have used 1000 mg of the potassium dichromate in the lab, and I have prepared my own solution, and I have done. I've not used the industrial effluent. So in the future work, we would go to use it for the effluents and find it out. And why I tried for 1000 mg per liter is because actually in the effluents, whatever it has been found till now is about 250 to 300 mg per liter is the maximum amount of the concentration we found in the effluents. But there are a lot of other substances which are also found, So if you try for 1000 mg, if they are efficient in 1000 mg, they can easily eliminate even this 250. So that was my aim. So I went with the higher concentration of the chromium. So this was my work. So any questions with this? So next slide. So this is the RSM design which I developed for 62 experiments. Next one. So these are the references which I referred. There are many more. I have just put some references here. Thank you for hearing. Any questions?
Thank you very much, ma'am. It was an excellent lecture.
Yes.
Whoever has questions, I would ask them to.
Yes, sir. Any questions? Please do ask.
Meanwhile, I will also type ma'am's email address in the chat box so others could follow her.
Yes, sir. Yes, sir. Please.
Good morning, ma'am.
Yeah, please.
Ma'am, I have doubt.
Hello.
Ma'am, in microtiter plate assay for biofilm formation,
Yes,
your
sir.
measuring OD value,
Yeah.
like more than one, it's a very good biofilm formed.
Yeah.
Did you check for percentage, ma'am? In at what range do you chosen for percentage in biofilm formation?
Yeah. Like what we've actually we measured the OD at 540 nanometers. So it was like whatever the OD we got from 0.2 to, if it was less than 0.2, it was not forming a very good film. Okay. So it was weaker, we found it out. So from 0.2 to 0.5 was medium, moderate type of fall. So above 0.5, what was there? So they were good ones. And we also did the tube assay to find out the amount of the film which was formed within the test tubes also. I had shown one blue colored test tubes, you had seen that, I suppose, with a crystal violet, right? So the once it was just for 24 hours and it is about only the 10 ml. Okay, in the 10 ml itself, you can see. And the biofilm producing organisms, whatever they are there, no? In 24 hours itself, you can see the layer, nice layer forming on the surface of the broth.
Okay. Whether it's taken in static condition or in shaking, ma'am?
It's better in the static condition, because what happens is if you keep it in the if it is not static condition, what happens is there will be like the shearing of the cells and you don't get the proper film.
Okay, ma'am. What about the Congo red plate method, ma'am?
Congo red plate method, actually I didn't get the proper results, so I missed the slide actually. So it formed the black color type of calling, it's actually red media, because you're adding the Congo red as an indicator there, and you have those if the organisms of biofilm forming organisms, they form the black color type of colonies, which is a good indication that they form the films.
Okay, ma'am. Actually, I'm working on biofilm, ma'am.
Okay, good.
So in previous study, I just used Congo red and then I skipped to tube method. While comparing these two methods, I get unrelevant results.
Even I didn't get proper Congo red results, so I relied on tube assay and ELISA plate method.
Okay, ma'am.
Okay.
Good afternoon, ma'am. Hello, ma'am.
Yes.
Good afternoon. Good afternoon, ma'am.
Good afternoon.
Ma'am, it's not a big query, but I want to confirm about I need a certificate of this webinar, of this today's webinar.
Okay.
Yes,
hello,
ma'am.
Dr. Madhumala. Thank you for your wonderful piece of work. You have tried to bioremediate 1,000 micrograms of hexavalent chromium. But my query, just a simple query is there. Actually, the biofilm is actually you have done the work with biofilm mediated bioremediation, isn't it? So it is absorbed on the biofilm. Is it absorbed on the biofilm as hexavalent chromium, isn't it? So after the death of the bacteria,
Yes, ma'am. It is.
what will be the fate of the chromium? The hexavalent chromium will remain as such in the soil, isn't it? So what is your suggestion?
No, ma'am. It is transformed into hexavalent, trivalent, because of these organisms. They undergo the redox reactions. Even the biotransformation also takes place along with the biosorption. So there will be transformation also, not only the biosorption, but transformation also takes place.
Madam, is there any definite or conformative reaction by which we can tell that it is being transformed from hexavalent to trivalent as such?
Ma'am, I've tried out in the liquid, like in the solution. I've used a synthetic solution of hexavalent chromium and I tried out and I have found it out. By diphenyl carbazide assay, you can find out the amount of the chromium we found within the media there, within that.
No, by dicarbazide method, we are measuring how much hexavalent chromium is getting reduced, but not in the form of trivalent as such. So my question is how this trivalent, how much trivalent is formed? Is there any confirmatory reaction?
There is one more method, ma'am, with the trivalent, but I've not found it out because what happens is normally the trivalent, it forms a precipitate in the liquid. So you can remove it. So suppose if you are going to go for effluent treatment and that it leaves as, it is normally found as a residue at the bottom. So you can easily remove.
Okay, but that's not a confirmatory one, isn't it? Okay, thank you.
Yeah, but there is one more test, ma'am. I don't remember it exactly now, but there is a test where you can find out the amount of the trivalent chromium also. You can mail me. I can send you that. I have that, but I don't have it at present.
Okay, sure. Thank you.
Sir, you can share my mail ID so that if they have any queries, they can mail. There's no problem.
I already have. I have shared her email address in the chat box. So anybody who might have other questions, please follow it through with her. Thank you very much, ma'am. Thank you.
Thank you, sir.
Thank you.
Thank you, sir. Deshmukh, sir.
Thank you.
There was a lot of issues. I suppose I'm sorry for that.
Okay, but after all,
sometimes we have circumstances beyond our control, but it was an excellent lecture. Thank you.
After all, the presentation was excellent. Thank you, ma'am.
Thank you.
Thank you.
Thank you.